Hello, everybody, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Garbage In equals Garbage Out, Complex Biological Sample Preparation for Omics Studies. Omics technologies can provide scientists with lots of useful information for any number of research projects involving new drug discovery, biomarker identification, forensics, and vaccine development, just to name a few. However, meaningful outcomes depend on careful sample preparation and analysis. Otherwise, the results just may reflect the process of garbage in, garbage out. Today's webinar has been designed to help you avoid going down this path. Our two panelists will discuss and demonstrate improved techniques for preparing and analyzing samples for proteomic and lipidomic studies. Let's meet our panelists. Dr. Alexander Lazarev is Vice President of R&D for Pressure Biosciences. Alex is going to describe and go into detail on the best approaches for preparing complex biological samples for omics research. Dr. Ivona Strug, Senior Biochemist at EMD Millipore, will elaborate on some of the points Alex addresses, as well as providing additional insights on sample prep and analysis. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as moderator. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many questions as possible. So if everyone's ready, let's get going. Dr. Alexander Lazarev will be our lead-off speaker. Alex, the floor is all yours.
Thank you, John. The topic of biological sample preparation is vast and most certainly cannot be adequately covered in a short webinar. Our goal today is to discuss the general approaches and introduce the audience to several successful examples and new trends in omics sample preparation. In the past several decades, enormous investments were made to achieve unprecedented resolution and sensitivity of analytical instrumentation for life science research. However, only in recent years, biological sample preparation had attracted attention of research groups and life science tools companies, resulting in successful development of a broad range of tools. As our analytical capabilities continue to improve, however, new challenges in sample preparation are being uncovered. As Wikipedia states, the neologism omics refers to a field of study in biology ending in omics, such as genomics, proteomics, or metabolomics. These fields study particular types of analytes collectively. For example, genomic studies nucleic acids and was initially focused on transcription profiling. Recent genomics renaissance is centered around next-generation sequencing. Proteomic studies, proteins and protein conjugates with other molecules, glycans, lipids, etc. Metabolomics generally covers small water-soluble molecules. Lipidomics is looking at non-water-soluble species, excluding large insoluble biopolymers. Glycomics may or may not be considered a subset of proteomics, although from the mass spectrometry and data analysis perspective, there are very clear distinctions between these two fields. There are many other less fundamental omics terms that were coined recently. However, as one popular joke states, although it's not linguistically related, the most important omics field that enables all areas of research is economics. But seriously, there's one more important point that relates to many omics techniques. Most analytical workflows require true solutions of analytes as input. However, biological samples are complex macromolecular assemblies of components that are compartmentalized by insoluble barriers such as biological membranes, cell walls, etc. This brings us to the topic of today's discussion. We are going to talk about biological sample preparation. In all omics techniques, quantitative recoveries and reproducibility are essential to achieve a desired experimental goal. Sample preparation for historically first omics field, namely genomics, as well as for modern next-generation sequencing, relies on the fact that nucleic acids possess fairly uniform physical and chemical properties and can be easily treated as a distinct group. Moreover, nucleic acids are easily soluble in water and precipitate in organic solvents. They also specifically bind to chromatography support materials such as silica Therefore, lots of commercial sample preparation kits are available today that utilize those properties of nucleic acids very well. Proteins, on the other hand, are very diverse and at present present in biological systems in a very broad range of concentrations. Low abundance proteins cannot be amplified as nucleic acids. Therefore, the optimal choice of sample preparation in proteomics heavily depends on sample type and chosen downstream analytical workflow. For example, in its early days, proteomics focused on biological fluids such as plasma and serum, where solubility was generally not a big problem. Perhaps the biggest issue for these types of samples have to do with the broad dynamic range of protein concentrations. For instance, the 10 most abundant proteins represent approximately 90% of total protein mass in human plasma. The 22 most abundant proteins are said to represent about 99% of the total protein mass. Unfortunately, most of those abundant proteins do not serve well as disease biomarkers, as their concentration is continuously affected by many factors, even in the healthy individuals. The most interesting part of the plasma proteome is hiding in that remaining 1% of total protein. Therefore, depletion protocols have been suggested as a solution to address the dynamic range problems by removing anywhere from 2 to 20 most abundant proteins from plasma. Traditional approaches for abundant protein depletion use mimetic dyes such as Cibacron blue or protein A or protein G to remove albumin and IgGs, respectively. While these approaches worked across multiple species, low specificity of these methods and a desire to remove a broader range of abundant proteins led to a development of immunoaffinity-based approaches. Antibody-based products are available for up to 20 human plasma proteins and several proteins from other mammalian species. Such products are offered in multiple formats, such as flow-through HPLC columns, spin columns, and magnetic beads. Generally, chromatography columns are suitable for routine work on a particular specific sample type. They tend to be pricey and require tender loving care to ensure long performance and no sample carryover. Spin columns and magnetic beads, on the other hand, are more forgiving and more versatile for small pilot studies and a variety of different samples. They also tend to be disposable. It must be kept in mind, however, that cross-reactivity between different species is typically very low, so specific antibodies must be developed for studies in animal models versus human patients. The bottom panel on this slide shows one of my personal favorites, a recent versatile product called Amicon Pro, offered by EMD Millipore, that combines abundant protein depletion or enrichment and ultrafiltration-based protein concentration in one convenient disposable format. What about plant samples? Rubisco, or ribulose 1,5-bisphosphate carboxylase oxygenase enzyme, is the most abundant protein in plants and possibly the most abundant protein on our planet. Several companies are now offering Rubisco depletion kits based on antibodies or specific precipitation methods. As shown in this example, the large and small subunits of Rubisco annotated with white arrows on the left panel of the slide appear to be the predominant features in the raw spinach leaf protein extract. There is little or no visible Rubisco bands in the depleted spinach protein fraction shown on the right panel. Even when we deal with human plasma and have access to a wide spectrum of commercial depletion kits available in the market today, removal of several most abundant proteins only gives us about 1 or 2 orders of magnitude of improvement in concentration. However, the most interesting biomarkers, such as some of the tissue-derived signal transduction proteins, appear at much lower concentrations in most plasma samples. Enrichment for those proteins of interest has been proven in many cases to be a successful strategy to solve that issue. Phosphorylation, for example, is the most popular post-translational modification in proteomics due to its role in signal transduction. Current methods to enrich samples for phosphorylated proteins are based on affinity purification using phosphospecific antibodies, immobilized metal affinity chromatography, in short, IMAC, strong cation exchange chromatography, or titanium dioxide chromatography. Anti-phosphotyrosine antibodies have been proven very successful in purification of phosphorylated proteins and peptides, but fewer reports have been published that successfully used antibodies against phosphoserine or phosphothreonine-containing proteins, since the affinities for those phosphorylated amino acids of different antibody-based peptide enrichment is considered inferior to other methods at the moment. IMAC enrichment, being the most popular approach, is based on phosphate affinity for immobilized metal ions chelated to the resin. Titanium dioxide chromatography is a newer technique that requires significantly less column preparation time. Strong cation exchange separates phosphorylated from non-phosphorylated peptides based on the presence of negatively charged phosphate groups. Many phosphoproteomic studies use the combination of these enrichment strategies to obtain the purest sample possible. Although currently not as popular, there are methods to enrich for glycosylated proteins or proteins carrying other specific post-translational modifications. Moreover, enrichment strategies are becoming even more significant for targeted proteomic workflows as proteomic enters its clinical phase. As custom antibody development becomes more affordable and reasonably fast, more antibody-based product kits are entering the market. Methods of abundant protein depletion and enrichment for specific types of proteins, including proteomes of organelles such as mitochondria, have been developed for tissue proteomics. This classic example from Professor Jennifer von Eyck's laboratory shows two-dimensional gels of whole heart muscle tissue lysate on the left side compared to the sample enriched for mitochondrial proteome from the same tissue. Organelles could be isolated from the tissue samples intact and then lysed in the appropriate buffer to produce nearly pure mitochondrial proteome. While many laboratories are still pursuing biomarker discovery in biological fluids, a recent paradigm shift to tissue proteomics presents new challenges for sample preparation methods such as solubility, stability, and a few other issues. Protein dissolution requires detergents and chaotropes that interfere with downstream analysis. In addition, there are many more challenges related to the real-world samples, such as lysis of particularly tough tissues, for example, bone, hair, skin samples, etc., several resilient species of bacteria, plant tissues, as well as protein extracted from complex matrices, such as food, soil, or fecal material, and so on. Even though mammalian tissue cultures and soft tissue samples are relatively easy to lyse in chemical lysis buffers, the use of salts and detergent necessary for such lysis steps presents significant problems with enzymatic digestion, chromatography, and mass spectrometry downstream. Those chemicals generally have to be removed soon after lysis. Ultrafiltration has been used for many years to replace buffers and remove detergents from protein samples. However, removal of detergents and chaotropes that were used to keep proteins in solution in the first place may lead to loss of proteins due to precipitation and aggregation. A creative approach termed FASP, developed in the Matthias Mann's group at the Max Planck Institute in Germany, relies on the use of strong detergents such as SDS to solubilize the entire tissue proteome, ultrafiltration spin columns to remove detergents and capture proteins on the membrane, followed by digestion directly on the membrane to access precipitated and aggregated protein species and turn them into soluble peptides. However, this approach is also not always ideal, since some aggregated proteins become very resistant to tryptic digestion and may not be fully recovered. Small tissue samples, such as needle biopsies of 1 to 5 milligrams, require special approaches for homogenization. Since these samples are so small, potential analyte loss due to binding to the container walls requires minimal amount of sample manipulation. Larger dilution factors are not desired as well, since protein losses to various surfaces are typically greater at lower concentrations. Also, since the total amount of protein in the samples is so small, depletion or enrichment strategies typically may not produce desired benefits with respect to assay sensitivity. One of the possible solutions for such small samples is to perform the entire sample preparation process in a single container and remove interfering compounds, leaving the useful portion of the sample in. A product from our company called MicroPestle is a small sample container that contains integrated homogenizer, a Teflon pestle that fits tightly into the tube after the tissue sample has been placed into it. The tube then is placed into an instrument where hydrostatic pressure collapses the container walls against the pestle to mash the tissue sample inside. Then the pestle is removed and replaced with a shorter cap, allowing the user to add extraction and digestion reagents sequentially directly into the same container. Minimal sample transfer results in minimal losses of protein. Challenges with proteomic sample preparations are not limited to protein dissolution and enrichment. The next step is proteolytic digestion, typically performed by site-specific serine proteases such as trypsin and the proteinase Lysate and several other enzymes, or their combinations. The digestion step typically takes a long time, as the enzyme is expected to quantitatively cleave all possible sites on all substrate proteins. However, the reality is far from ideal. Digestion kinetics is substrate protein specific. Therefore, digestion step becomes a time-consuming process. Moreover, some proteins are very resistant to proteases and therefore may escape complete digestion by conventional proteomic methods. Several approaches have been suggested to accelerate protein digestion. Among those, there are chemical additives such as acid labile detergents and physical methods such as microwave heat, ultrasonic mixing, or incubation at high pressure that help to keep substrate proteins prepared for digestion. One must always keep in mind, though, that enzymes such as trypsin are also proteins, albeit a bit more stable than most substrate proteins of interest. So care must be taken to avoid inactivation of protease activity in an effort to facilitate denaturation of substrate proteins. An instrument recently developed in our laboratory called Barozyme HT48 is specifically designed for proteomic and glycomic enzymatic digestion and accommodates up to 48 samples per batch in the microtiter plate compatible format. Hydrostatic pressure is used in this instrument to pre-denature protein substrates but keep trypsin intact and active. In this example, unreduced and unalkylated samples of monoclonal antibodies were digested with endoproteinase Lysate under alternating hydrostatic pressure, as compared to the overnight incubation and the 3-hour treatment by microwave radiation at ambient pressure. High-pressure treatment in only half an hour tends to produce less miscleavages and higher signal intensities of resulting peptides, while leaving less incompletely digested material behind. Recent work from Professor Rudi Ebersold's laboratory at the ETH Zurich in Switzerland introduces a new workflow entitled PCT swath. Small tissue biopsy samples are extracted and digested under pressure cycling conditions, while sample material never leaves the sample container. This method minimizes potential of analyte loss while maximizing solubility of membrane proteins, digestion efficiency, and sequence coverage. The lossless data-independent MSMS data acquisition method called swath-MS enables collection and storage of data for all detected peptide fragments for later use, while targeted data extraction offers rapid quantitation of peptides of immediate concern. This approach intends to produce a digital snapshot of each sample for future data mining efforts. Needless to say that the quality of sample preparation for such workflow is expected to impact the successful future discoveries. Immobilized enzymes promise to simplify digestion and offer automation of protein digestion steps. If enzyme is covalently attached to a chromatography resin, it could be placed into a stream of mobile phase and sample could be loaded into such column automatically using column switching valves available on most chromatography workstations. This approach accelerates digestion by offering a very high local concentration of enzyme on the column. It also minimizes self-digestion of the enzyme, which cannot reach each other while being immobilized, but extreme care must be taken to avoid sample carryover or inactivation of enzyme by passing high concentration of organic solvent. Immobilized trypsin columns are now commercially available from several vendors, although they are relatively expensive and eventually require replacement. Traditional methods intended to extract a distinct class of analytes may not be able to preserve other types of small molecules Traditional methods intended to extract a distinct class of analytes may not be able to preserve other types of sample molecules, or even fail to capture the entire class of desired analyte molecules, leading to an inevitable sacrifice in sensitivity, or a need to use orthogonal methods to prepare multiple sample aliquots for specific workflows. As omics methods enter clinical applications, the demand for working with small samples like tissue biopsies require optimization of sample preparation methods towards multi-analyte approach. Ideally, a true sample fractionation would be needed to separate water-soluble metabolites, lipids, and proteins into distinct fractions from the same small sample, based on perhaps different solubility in various solvent systems. However, current multi-omics workflows, which you see in the literature, frequently share only a common cell tissue lysis step. Afterwards, individual aliquots of the lysate are treated independently for their distinct omic techniques. In addition to sample preparation issues, lipids, for instance, present a new set of challenges. There are no straightforward way to identify a lipid molecule since there are no genes that directly encode lipid structures as they do for proteins. Moreover, many lipid species are isobaric, meaning having the same molecular weight, positional isomers that possess distinct biological functions in the cell and therefore need to be quantified separately. Until recently, mass spectrometers could not easily distinguish between isobaric species, so high chromatographic resolution and long HPLC gradient runs were required for successful lipid profiling. With the introduction of ion mobility mass spectrometry that separates ions by their shape rather than mass, we gain a new dimension, high-resolution separations of lipids that can rapidly distinguish between species with identical molecular weight. This example from Dr. Arthur Mosley's lab at the Duke University Proteomics Core nicely illustrates the multidimensional ion mobility separation of complex lipid profile extracted from tissue culture samples. Lipidomics is an emerging field that has immediately attracted attention of clinicians. Our collaboration with several groups at Harvard Medical School, Brigham and Women's Hospital, and Mass General Hospital has been focused on development of lipidomic analysis of neonatal fecal material, the workflow that promises to offer opportunities of early diagnosis of improper bacterial colonization of infant gastrointestinal tract and other digestive disorders in premature infants. Disease called necrotizing enterocolitis, NEC, is potentially life-threatening for infants with digestive system that is not fully developed yet to accept oral nutrition. In this work, we have compared popular modified Bly-Dyer lipid extraction method and alternative solvent system consisting of the fluorinated alcohol hexafluoroisopropanol and methyl tert-butyl ether with and without the use of high hydrostatic pressure to facilitate extraction. Both solvent systems under pressure extract a significantly higher number of lipid species from infant fecal samples for analysis in positive and negative ion modes on the mass spectrometer. Analysis was performed using the thermo-exactive mass spectrometer in both positive and negative ion modes. Data analysis was done using customized thermo-sieve software. The study shows that diversity of lipid species in complex samples such as fecal materials require more than one parallel method of sample preparation to capture all potentially interesting analytes. This concludes my part of today's webinar. I hope I was able to convince you that successful sample preparation lays the foundation for subsequent analysis in omics fields, particularly in proteomics and lipidomics. Thank you, and back to you, John.
Alex, thank you for that in-depth look at the various approaches for preparing biological samples for omics research. Much appreciated. Wonderful presentation. Thank you. If you're just now joining our webinar, a big welcome. As I mentioned earlier, we will be conducting a Q&A segment following the panelists' presentations. Please type your question for any or both of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Dr. Ivona Strug, is ready to begin her presentation. So, Ivona, I'm turning everything over to you.
Thank you, John. And thank you, Alex, for such a broad description of methods and problems involved in sample preparation connected to various omic studies. I hope to complement what Alex has just presented with short introduction of recently introduced method for sample qualification. So let me start from the very short and very brief summary of current protein quantitation methods. All methods are relying on the electromagnetic vibration. There are methods connected to ultraviolet absorbance. In UV, there is 2AT for protein quantitation, and there is amino acid analysis. All colorimetric assays are placed in the visible range of spectrum. And as I said, the most recently introduced quantitation methods for biologists is based in IR space. Why IR? Well, we have decided that it would be nice to base the quantitation on the vibration of the functional groups. If you look at the protein, the amide bond is dominating the structure because the peptide bond is the main structure of the protein. There are other biomolecules, like, for example, the lipids or detergents, which have the characteristic structure, characteristic bonds, like, for example, aliphatic compounds. So basing the quantitation in the IR area, protein quantitation can be done, but also other information can be delivered on the sample. On the next slide, I am not trying to introduce the product. I'm just summarizing what the new system can do. First of all, to use the IR qualification system, minimal learning is required. Nobody has to be IR expert to use this system. There is minimal to none sample preparation. The sample required for single analysis is only 2 microliters. There is no assay involved. The sample can be used in the form in which it was prepared. Also, the system uses the calibration, and the quantitation is based on calibration. However, the calibration can be done once for the buffer and stored and reused. Also, one additional thing about the system is that it's pretty much a shoebox. So any space restriction present in the lab, it's not applicable to this system. Why IR application and what's new in it? IR application for the protein quantitation, protein analysis, and so on, it's nothing new. Protein analysis by mid-IR is as old as 1950s, and a lot of publication can be found. Last decade, improvement of the instrumentation led to the quantitation by IR. However, all the approaches available until this system has been introduced required quite high IR expertise. What's new and why we can provide the IR quantitation to non-IR experts? It's a membrane. The possibility to place the sample on the membrane, which is almost invisible in the IR spectrum. If you look at the slide, there is a spectrum of the membrane there. It has the strong signal, but this strong signal is to the right of the absorption of both bonds which we are using for quantitation. So what is required from the user to analyze the sample to quantify the protein? The user needs to have a buffer in which the sample is prepared and the sample. The buffer is spot on one position, the sample is spot on the other position, or as many of us, it's using in triplicate on positions 2 to 4 of the card. Then the card is inserted to the instrument. The information about the sample is delivered to the software, and the measure button is clicked. Within a couple minutes, the instrument will dry the sample and deliver the dual results. Why I'm saying dual? Because numeric information will be delivered and spectral information will be delivered. Both are important for validation of the results, and I will be talking about it in the moment. Let's start from very important information, and I mean the fact that our analysis is not concentration, it's mass dependent. The sample is spot on the cart, dried, and analysis is performed on dried sample. So we are asking to spot 2 microliters and exactly 2 microliters. Spotting less or more, will be leading to the errors and inaccuracies in the quantitation. What additional things are connected to our analysis? Any water-based sample spotted on any hydrophilic material is drying, forming so-called coffee ring. To retain the material, the sample within the certain area, the embossing ring has been added to the sample spot. However, if you see on the slide, the same amount of the cytochrome C, which was used in this visualization only for the purpose of its color, were spotted from different buffers. And all 3 pictures show how differently it's dried on the sample spot. It is pretty much why instrument calibration from each buffer is required. However, once made such a calibration, it's reusable. Coffee ring effect is also connected to the characteristic of IR beam. With the current design of the IR instrumentation, the beam, it's not strong. It's not showing the strength across the sides. It's the most intense in the middle, and the intensity is decreasing toward the edges. So when you think about coffee ring, pushing the sample to the edges and the IR beam being the most intense in the middle, you see that those 2 are going in the opposite directions, resulting. If you look at the signal delivered by the cytochrome C spotted from water and cytochrome C spotted from 2 different buffers, one containing salt, the other containing detergent, which broke the water tension, which was pushing the cytochrome to the edges, the signal strength is differing by half. It's the other reason why the calibration from given buffer is so important for the proper quantitation. However, how the data is analyzed, it's also important. We start from collecting, as I said, the buffer spectra. Here in this spectrum, in this slide, the buffer spectrum is blue. And then we are collecting the sample spectrum. I'm showing the full spectrum for mid-IR range. When the analysis is performed, only part of the spectra is used. For example, if the protein analysis is performed, the buffer signal is subtracted from the protein signal only in the region which is important for protein, resulting in, let's say here, red spectrum. In case of lipid on detergent, the same operation is performed, but just in different area of the spectrum, resulting in the green spectrum, which is processed further, and I will talk about it in a moment. When the spectrum is obtained, buffer is subtracted, then the specialized software can be used with the Calibration Wizard to lead the user to generate in generation of the calibration method. There are 3 steps. One step, the user is choosing the calibration method. In the second, it's choosing the files with the spectra and assigning concentration to given spectrum. And in the third step, the calibration is the spectra are integrated and calibration curve is generated. There is possibility to go back and forth between step 3 and 2 until the satisfactory calibration method is generated. As I said, we are mostly oriented toward the protein quantitation. There are 9 bands characteristic to the proteins in IR spectrum. However, the one which are underlined here, amide1 and amide2, have been identified as the most representative and they were most studied. So we use both. However, amide2 is used only for visual purpose, so it's only in the display. All analysis quantitation is done on amide1. I would like to underline one thing here. When we are quantifying proteins, we should trust the data, and we have to know if we can trust the data. Here is an example where the various proteins were used to generate the calibration curve for MIR method and for the UV280. As you can easily see, the calibration by various with various proteins in MIR gave quite overlapping results, while the differences in tryptophan, tyrosine, and cysteine content of the proteins led to a UV280 to deliver very distinct slopes. I could put the other comparison here. My goal is not to discourage anyone from using other methods. My goal here is to show that whatever protein is used to calibrate using MIR-based technique, it's good. We also acknowledge also a couple other things which are connected with the protein quantification by mid-IR. First of all, if you go and check the information on the location of amide band in amide1 band, in the IR spectrum, you can see that the maxima can shift substantially depending on secondary structure. Amide1 signal will always show between 16 and 1700. The difference is where the maximum will show. To account for this, there is possibility to build the calibration method, which is hooking a baseline between 16 and 1700, and then measuring the highest, finding the highest point and measuring intensity at the highest point. For this reason, when using this type of integration, any protein can be used for instrument calibration. However, since we are using the amide bond signal, and within the amide bond signal, we are going after the vibration of COO and CN bonds, it is easy to imagine that there will be buffer components which will contain those one or both of the bonds, and those components might absorb in IR system the same way as the amide bond. Here I'm giving, in the slide, I'm giving the example of quantitation from tris buffer. Tris contains CN bond, So it will absorb within the amide-1. The top spectrum is showing the trace of absorption pattern, and the bottom spectrum is showing the trace absorption and the protein absorption, so pretty much protein in the trace buffer. Please don't pay attention to the fact that the spectra are separated. It's done artificially. If both were run as they were collected, they would overlap. The major point of this slide is to show that quantitation in this case is also possible. It requires slightly different approach to spectra integration. So in this case, the baseline is again hooked between 17 and 1600, but at the same time, the quantitation, the signal height, it's measured at the predetermined wave number. For this reason, the calibration from such a buffer has to be done with the protein which is structurally similar. So for example, if I'm using the BSA to calibrate, I should quantitate the proteins which have dominance of alpha helical structure, not the antibody. If I want to quantify antibody which has dominance of the beta sheeted structure, I should calibrate with the well-characterized antibody. There is another possibility. I'm giving another example of the acetate, for example, which contains C-double-O bond. In this case, the buffer doesn't absorb within the amide-1 region. It absorbs outside but encroaches the amide-1 region. For this reason, if the baseline was hooked between 16 and 1700, the protein signal would have been lost completely. So what the method does, it attaches the baseline at the predetermined wave number outside the analysis box and runs the baseline in parallel to x-axis and measures concentration at, again, predetermined wave number. Again, as with the previous option, we recommend to calibrate the instrument with the structurally similar protein because the determination is made at the predetermined wave number. How accurate is the measurement with the system, and where is the dynamic range? Since we simplified the analysis, we sacrificed to a certain degree the lower limit of quantitation. With this approach to the data analysis, all spectra below 0.25 mg per ml are too noisy to be reliable. So the lowest limit for this analysis is 0.25 mg per ml. We recommend building the curve up to 5 mg per ml. However, this limit is not absolute. There are examples of protein which can be linear up to 50 mg per ml as protein A, or up to 35 mg per ml as the lysozyme. However, at the same time, there are proteins which will start to lose the linearity of the signal increasing with the concentration, like, for example, BSA. Also, the instrument is capable of quantifying delivering spectra, which will be quantifying protein at the ELISA level. The one thing which has to be done to such a spectra is complicated analysis, which would include chemometrics. So for all of those who are savvy with IR data processing, the data can be collected on this system, transferred to some IR software, and processed. As I said before, we concentrated on protein quantitation, but it doesn't mean that IR cannot do more. Here is an example. the system can go after aliphatic components because the data is already collected. And this aliphatic component has been used to prepare the method that would allow lipid or detergent analysis. During the development of the method, we found out that depending on the aliphatic content, the given analyte will deliver different calibration slope, which led to the finding that unfortunately, if using aliphatic component, there is no universal standard which can be used for the instrument quantitation and universal use. For reliable quantitation, the instrument has to be calibrated using the lipid or detergent which is further analyzed, or mixture of lipid and detergents at the same ratios as in the analyzed sample. Said what I just said, my friend running one of the mass spec facilities in the area was having troubles with lipidomic studies because from this, what I was told, her method of quantitation of the sample led to instrument overloads or injection of not enough of the sample. What she did is she bought the total liver extract from Avanti Lipids, used it to calibrate direct detect, and with this calibration, she didn't have the quantification method, but she claims that she developed the qualification method, which allowed her to enhance throughput because she was finally injecting the amount of the sample which was sufficient to obtain the data she was looking for. What the module of the lipid module allows also for. We are collecting the full MIR spectrum. So the spectra between 400 and 4,000 of it's collected. This spectra contains information about protein, information about lipids, contains also additional information which can be extracted not quantitatively, but it can help to analyze the sample and can qualify the sample throughout the processing. I give an example. In the very short breast cancer protein biomarker studies, we use two different lysis buffers, wanting to know how much of lipid and protein we are extracting and how many spins of the additional manipulation has to be done to the sample before the sample can be analyzed for the biomarkers. Please don't pay too much attention to what's on the slide. I'm just showing here the schematic how the sample was processing. What's important is information on the lipid and information on the protein, which were obtained in one single analysis. For example, when we were monitoring lipid removal, we wanted the protein which is lipid-free. So looking at the process, we were getting a couple of information just from the analysis in the system. First of all, one buffer was recovering more lipid than the other. And within 2 spins, 3 spins, we were able to get lipid to the level which was almost at the edge of the system detection. When it comes to the protein, we got the information that, first of all, 2 different buffers were recovering different amounts of protein, and one buffer was leaving quite a bit of protein behind in the lipid fraction. So what the system, MIR-based system, can do? It can quantify the protein, but also deliver additional sample information. It uses the sample which is not processed. There is no assay involved. The sample is spotted on the card. The card is inserted to the instrument. Spectra is collected for the buffer. Spectra is collected for the sample. Data is processed automatically, and the answer is delivered within a couple minutes. It is pretty much what I prepared for today. This slide concludes my presentation. Thank you very much for your attention, and back to you, John.
And thank you, Ivona, for that very detailed presentation. You and Alex have provided our webinar attendees with some excellent ideas and suggestions for improving their sample prep procedures for omic studies and avoiding a potentially disastrous garbage in, garbage out scenario. We're almost ready to begin our Q&A session, but first I'd like to ask you to disable your pop-up blockers because a short survey on this webinar will be appearing in a moment. We would very much appreciate your feedback on this webinar. Okay, we have our first question, so let's get the Q&A session going. Alex, we have a first question for you. What specific problems are posed by specimens that had been formalin fixed both for downstream proteomic and genomic analysis?
Thanks, John. It's a very good question, and in fact, I would like to combine it with the following question, number 22, I guess, which relates to paraformaldehyde cross-linking. The problems of tissue fixations are mostly related to the fact that it's a chemical cross-linking of all the molecules in the sample. And the biggest issue is to actually reverse the cross-linking to release the molecules, whether proteins or DNA. In particular, all of these things get really tied up with covalent bonds. And the classic approach to releasing of proteins from such specimen was the use of temperature. High temperature is shown to reverse the cross-linking reaction and release the analytes. The problems are as follows: that most of the fixed samples stored for indefinite period of time, sometimes very long. And during the storage and during the decross-linking process, there are chemical modifications which do occur to some fragile residues on the protein molecules, for instance, or on DNA. And those modifications will have to be taken into account during the informatics data analysis, even if cross-linking is accomplished quite successfully. I want to bring up the example of commercial kits. Original method was commercialized by the company called Expression Pathology. Their kit called Liquid Tissue is still quite popular in reversal of the formalin cross-linking for protein extraction. And since then, the variations on the same theme have been published many times with slightly different chemistry used. But they all use high temperature, and temperature is absolutely crucial to reverse the cross-linking. In addition, the methods have been suggested to essentially capture the free formaldehyde, which is released during the decross-linking process by scavenging it with some other buffer components, abundant amine-like functionality, which would bind to it and prevent the recross-linking of the macromolecules.
Okay, you want to tie that at 22, so let me read 22. Do you have any advice for extracting protein from PFA-treated tissue that allows me to quantify the protein after and use the sample in an ELISA? That is, can't use anything that would affect antibodies.
Yes, so paraformaldehyde is very similar to formalin in its ability to cross-link the molecules. However, it's known to be a less active cross-linking, so it's a shorter molecule and it doesn't cross-link as efficiently as formaldehyde. So you have more luck in recovering some of the proteins, in particular from paraformaldehyde-treated tissues. But same challenges remain. Depending on how long the specimen was stored, you have some modifications happening, and also decross-linking has to occur, and the temperature is the best bet case. So the removal of some of these chemicals used in decross-linking protocols could be done, especially if you're trying to recover some proteins. Ultrafiltration would be a great example. If you're going with the proteomic methods and want to digest the samples and run the mass spectrometry, that people have successfully used the FASP technique, which I mentioned about in my slides, which uses up to 4% SDS to solubilize remaining material after decross-linking and then remove the detergent and digest the things on the membrane. So again, ultrafiltration and Millipore has been offering great products in the consumable format with ultrafiltration membranes, a great way to remove the interfering compounds before you use your antibodies for ELISA or any other immunoaffinity methods.
And Alice got another question. And first of all, somebody says, thank you for a very informative talk. Is there any method for enriching signaling molecules and transcription factors from the total protein lysate?
It's a very important question. However, the answer may not be as simple as it may sound. Signaling molecules range from simple gas molecules like nitrous oxide to large proteins. And obviously for particular classes of signal molecules, the methods have to be very specific. And I guess since we are focusing on proteins and lipids in this particular talk, on the webinar, I would cover the proteins. And as I mentioned, phosphorylation has been worked on for a number of years, and there are commercial products and published academic work on enriching for phosphorylated proteins and phosphorylated peptides. In fact, most laboratories, as far as I understand, prefer enriching for phosphopeptides after digestion. Although one has to deal with the initial sample complexity before the phosphopeptides are produced. But in other way, let's say transcription factors, they're known to bind to DNA, and so the chromatin immunoprecipitation methods and similar methods to that are used to use DNA as a template to bind those specific transcription factors out of your total lysate, and then precipitate them along with DNA to enrich for those. And again, this could be a topic of a major discussion on itself. But in many ways, unfortunately, there's no specific method to enrich for all signal molecules, and we'll have to treat them all as specific classes based on what they do in the cell and how they could be captured.
Well, thank you very much. Ivona, question for you. I used direct detect to measure the concentration of a lyophilized peptide that I reconstituted in buffer, and the results showed that the peptide concentration was lower than what I expected. Can you explain that? Ivona, are you there?
Yes, I am.
Okay.
I hope I can answer this question, and I think I can answer this question. Let me start from statement that since we developed and launched the protein quantification system, we've encountered quite a lot of questions about peptide quantification. What happens is this instrument is very sensitive when it comes to the quantitation of peptides. And what I mean by very sensitive is that it will pick up peptide impurities. If the peptide is super pure, the system will quantify it really correctly. However, if the peptide carries impurities, the system will show not one signal within analysis region. It usually shows 2 or 3 peaks within analysis region. And then the system is getting confused which of the signals it should quantify, it usually will choose, by default, it will choose the highest signal, or if you are using analysis method 2 or 3, it will use 1658, the wave number. So the system will allow quantification, but only if the peptide is pure. So my assumption in this case is that this peptide wasn't pure, and system was just checking the amide bond or the highest signal.
Okay, we have another question, Ivona. Since direct detect can be used to measure aliphatic chains in proteins at the same time, has anyone used it to analyze extracellular vesicles?
Yes, customers did. There was a poster at one of the conferences in Rome a couple months ago, and we did. The challenge to analyze extracellular vesicles is that usually they are co-purifying with albumin, regardless if the purification is attempted from the cell culture, media, or any of the body fluids, including urine. So since direct detect is seeing total protein, whatever protein which is co-purifying with the extracellular vesicle will be also quantified.
Thank you very much. And question, could you explain a little more about what proteins or samples might be hard to quantify using a Bradford assay?
I do think that there is plenty of literature on this subject. I can provide, upon request, the papers. The problem with Bradford assay is it is dye-based. So dye is binding to the protein, and Bradford is using Kumasi, which is looking for the amines in the protein. So let's assume that protein the BSA is used as the standard to develop a standard curve. And with this standard curve, I can quantify properly only the protein which has the same amount of lysine and arginines which are accessible. Also, in addition, there is a paper outlining very nicely that the dye binding affinity to the protein depends from vendor to vendor. In this paper which I'm talking about, the authors compared two different vendors. The dye from one vendor was strongly underestimating the concentration of low molecular weight proteins. The dye from the other vendor was underestimating the concentration of high molecular weight proteins. So my personal recommendation, I never go close to Bradford. I just don't trust Bradford. If I have to run colorimetric assay, and I'm underlining, if I have to run colorimetric assay, I would use BCA.
Okay, thank you. And another question, Ivona. Bear with me, it's a little long. I'm attempting to run an experiment in which I gain both a metabolomic and lipidomic samples. I'm using a chloroform methanol method, which results in a white cell debris layer between the phases. Can I gain proteome analysis from this white layer?
Proteome analysis, meaning total amount of proteins, I guess in the estimation. Our membrane can hold pretty much every single organic solvent. It is compatible with the chloroform. What it was used there, chloroform and methanol. It works with both. So the sample can be spot. I assume the sample can be dried and then analyzed. The only issue which I would have with this is the transfer, because I do not believe that there is possibility to spot exactly to microliters, and I don't know what to recommend in this case as the the buffer, because everything is coming from the white layer. I assume that the layer is fluffy.
May I also contribute to answering this question, because it's dear to my heart. This is Alex. We have tried this, and it's actually possible. The problem is, and I refer to the further downstream analysis, such as digestion and mass spectrometry, to gain the composition of proteins. The challenges are that essentially precipitation of proteins in the conventional chloroform methanol Bly Dyer method is very harsh, and essentially the aggregation is quite strong. So one has to consider that and redissolve that before digestion can occur. And in order to address that question, we actually looked at comparison of this method with the one I briefly mentioned in our fecal lipidome study. Actually, method was initially developed for proteomic analysis, and that's why we used alternative solvents to keep the entire cellular content in solution using fluorinated alcohols, and then slowly use liquid-liquid extraction to pull the lipids out of that extract and keep the proteome in solution. And this seems to work a lot better in terms of recovery and consistent recovery of the proteins from the sample. Although I've seen several publications where people have used Bly Dyer and collected the protein pellet and then redissolved it using SDS and followed it with the FAST method to gain the sample for subsequent mass spectrometry analysis.
Okay, well, thank you for two perspectives on the question. Unfortunately, though, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. I want to say thanks again to the panel for the outstanding presentations, and a big thank you to our audience for your attention and for your thoughtful questions about various topics you all brought up during the webinar. And thank you to EMD Millipore, whose support made this webinar possible. Bye-bye for now.